Most of us are familiar with identifying these common mosquitoes, and Louisiana is great for mosquito, or for quinquefasciatus populations due to the open septic ditches that we have. It's just great to have these open, wide ditches full of organic material for the quinques to come in across their airbrowse. Now, monitoring resistance is important, mosquito resistance specifically, because they're big vectors of a lot of diseases. Dog heartworm is a very common quink disease, as well as West Nile virus and other cephalitis. The reason that we're identifying and we're doing resistance for insecticides, well, mosquito abatements around the state and in East Baton Rouge Parish use insecticides as control. Nalin and resmetirom are the two main insecticides in East Baton Rouge Parish. Nalin is an organophosphate with a phosphate ester, and resmetirom is a pyrethroid with a carboxyl ester. Now, don't get too caught up in the chemistry or anything, but this ester right here is very important in resistance in these mosquitoes, perhaps. And then resistance in general, why is it important? Well, we all know that eventually these mosquitoes become resistant and they become hard to kill, but a lot of us may not know how this happens. So I've got a bell curve here. This would be the concentration that we're administering to the counties, the parishes, for control, and then this would be the population. Most members of the population are going to fall into this category right here, this 50% between, well, 60-some percent between these two standard deviations. And when we apply pesticides, they're going to kill the majority of the population, but there's still going to be this little tail in here that are left. Well, genetically, all the susceptibles are gone, and they can no longer spread their susceptible information. So the only people who are breeding is this little resistant until they repopulate the area and the whole population is now resistant. And mosquitoes become resistant through three main mechanisms. The first one is reduced penetration. For whatever reason, the molecule is no longer able to attack the target site the way that it wants to. Maybe it's a thickened exoskeleton or a waxy layer or some mechanical reason why the chemical cannot reach the target site. The second two mechanisms are more important. The first one being a target site mutation. What happens here, whatever the target site is, a genetic mutation has caused it to change. So with some pyrethroids, there'll be a base pair switch that causes a physical change in that sodium channel. So now the pyrethroid is attacking that channel, it can't kill them as effectively as before. And the third mechanism, which is the one that I'm most interested in, is a metabolic mechanism. Basically, there are three big enzymes inside mosquitoes that can change a molecule in one way or another so that the mosquitoes are no longer able to die from it. Esterases are one of those enzymes, and they're the ones that I'm most interested in because most of the insecticides that I'm looking at have an ester. Well, for my project, there are three main objectives. The first thing I need to do is, of course, determine levels of resistance across East Baton Rouge Parish. If there's no resistance, then there's no study. Second, I need to investigate esterases as a detoxification mechanism. If it is this mechanism, there are ways to go around. We can combat it. We can use different insecticides, or we can use synergists, which is the third part of my project. I want to look at Nalin as a synergist to resmetirom. Nalin, as I said, is an organophosphate, but it's also an esterase inhibitor. So if esterases are the problem, then perhaps Nalin is the solution. To do this, I need mosquitoes. Harris County, Texas provided me with a Sebring strain, which I have reared and created a colony. And to feed them, on a kind of interesting note, I used this membrane system here, where I take a Rutledge chamber, I put a piece of paraffin over the bottom, fill it with blood, stick it on top, and then I run hot water through it to keep it at a certain temperature. Because it does seem the mosquitoes like the temperature more so than the type of blood or the attractants, because I do breathe over it to see if CO2 can bring it to it. Secondly, I need field collected mosquitoes. And to do this, I go out and use Baton Rouge Parish. There are two ways. And I either set gravid water, and I go and I collect individual egg grafts every day, or there are some pre-found sites that myself and the research associate in the medical entomology lab at LSU use, and we take, just a little scoop with a stick, and we pull up, we scoop it, and we get them, and we just separate the egg grafts, and then we rear them. And then to test resistance, I need to establish LC50s for both resmetirom and Nalin. And an LC50 is just the concentration that I would apply to a population to kill 50%. To do this, I use bottle bioassays. It's a modified protocol from WHO, which most of us are probably pretty familiar with. So, but for my nalid, I found that the bottle bioassays just weren't effective. It seemed that I put acetone in with the concentrations, and as quickly as the acetone would evaporate, so would the nalid. So I changed it, and I created this topical assay where I have a CO2 knockdown chamber right here with a vacuum filter funnel. It puts our CO2 up there, I put the mosquitoes on, they knock down. And then I take very gently with forceps, and I pick the mosquito up by their legs, and I take the syringe, which can dispense a concentration in 1 microliter volumes. So I push the little button, and I get a very tiny droplet at the end of it, and I apply that droplet on the abdomens of the queens, and then I throw them back in the vials, and I watch to make sure that all of them pop back up. And then to determine levels of resistance, instead of going through and creating these LC50s for every single one of my sample populations, it takes a lot of time, I've determined that I'm just going to use diagnostic concentration corresponding with an LC85. So if I know that at this concentration 85% of my susceptibles should die, how many of my field, what percentage of my field population are dying? And I can figure out and calculate levels based on that. Now, to determine my mechanism of resistance, I use a spectrophotonic assay using a Spectromax 190, and I take each mosquito, I weigh it, I sex it, I grind it up, and then I put it in this machine with a substrate, and I can actually measure color production over time. And based on that color production, I can determine the level, the concentration of esterases in the mosquito. Now, as far as results go, I've only got some preliminary results. I have established an LC50 for my resmethrin, in my Sebring strain, and that would correspond with about 680 nanomolar. And while I was kind of perfecting the bottle assay, I was using mosquitoes from one of my samples of Ministry of Love. So I was fortunate that I have a full line. And I can tell right here, or I extrapolated that the LC50 for this wild population is 3,160 nanomolar. So this Ministry of Love sample is about 4.6 times more resistant than my control susceptible Sebrings. For my esterase assays, I again use my Sebrings as a control, and I have mosquitoes that I've collected from my East Baton Rouge mosquito abatement and rodent control site. And, it's amazing. It looks as though, it's 2 times, the concentration of esterases in the wild mosquitoes are 2 times as high as control, which could suggest that, esterases is an amplification. It's a higher number of esterases as the mechanism. Now, I mentioned previously what this tells us. Well, the first thing we can do is we can stop using ester insecticides. There are plenty of mosquito adulticides out there registered that do not have this ester in them. So that right there is an easy solution, but it may not be cost effective. The second thing we can do is we can use synergists. Both of these insecticides are already on hand at East Baton Rouge mosquito abatement and rodent control, so it could be beneficial to them to use a sublethal, smaller concentration of the nalid and a sublethal, smaller concentration of that resmethrin, and perhaps they're going to get better control than they did just using the resmethrin alone. This work still has some more to do. I mentioned that it's probably an overproduction of esterases, but it could just be a different one altogether. I don't know yet. So I plan on doing some gel electrophoresis studies to determine if it's a different one or if it's just more. Also, I need to compare 7 more populations, and I still need to create my nalid resmethrin-resistant strains. By the end of this, what I would like to do is to be able to compare a wild population against a control and a known resistant. And then finally, I actually need to investigate nalid as a synergist, and I'll do this using my Sebrings once I have both LC50s for both lines established. Well, at this time, I'd like to thank you and open up the room for any questions. Please, Dr. Kramer.
In your topical assay, you said you applied the insecticide to the abdomen of the mosquito. Why just the abdomen? Why not the thorax, or does it make a difference?
It doesn't really make a difference. The abdomen was the easiest place for me to put the drop, but as soon as you put that 1 microliter volume on, you watch the liquid spread all over the entirety of the mosquito, with the exception of the wings and the legs.